Okay, figure 624. This is the first figure we look at in section C of chapter 6, which is the section of this chapter that specifically looks at the synapse. So this is an important section of this chapter because synapses are really where the action is in both the peripheral and central nervous system. Synapses are junctions that we've been talking about these, but synapses are junctions that occur between neurons, and they come in two main types. The first type are chemical synapses, where a neurotransmitter is released into a synaptic cleft by a presynaptic neuron, and it binds receptors on another neuron that's postsynaptic to it. You learned about presynaptic and postsynaptic neurons previously in figure 6.5 in section A of this chapter. The second type of synapse is an electrical synapse, where there are gap junctions that physically communicate across the synapse between a pre- and postsynaptic neuron or other excitable cell. These gap junctions transmit signals from the presynaptic cell directly across the synapse to the postsynaptic cell without the use of neurotransmitters. So communication between electrically excitable cells using electrical synapses is very rapid, and the vast majority of electrical synapses are found in cardiac cells and muscle cells, while the vast majority of synapses found in the central and peripheral nervous system are chemical synapses using neurotransmitters. So specifically, figure 6.24 here describes the concepts of divergence and convergence in the nervous system. In convergence, thousands of presynaptic neurons can synapse with a single postsynaptic neuron and communicate with it. So convergence allows information from many different parts of the brain to influence a single neuron's activity. So conversely, divergence, a single presynaptic neuron can synapse with several postsynaptic neurons. So this allows a single neuron to have influence on many other neurons within a small area of the central nervous system. As we will see soon, neurons need to sum up all of the information coming in from presynaptic neurons and decide whether to depolarize and initiate an action potential or not depolarize and start an action potential. Whether the neuron depolarizes or not depends largely on how many of the presynaptic neurons are releasing excitatory neurotransmitters and how many are releasing inhibitory neurotransmitters at any given moment. So we'll be looking at that very soon and figuring out exactly how it is that a neuron makes a decision in terms of the information it's receiving as to whether or not it's going to fire. Let's go to the next slide. Okay, take a look at figure 625 here, which is the diagram of a chemical synapse. Remember, the chemical synapses are the ones that are present in the brain, as opposed to electrical synapses that you would find in cardiac and smooth muscle cells. If you look at this thing, you can see an axon of a presynaptic neuron ending, and there's a space, and then there's a postsynaptic neuron. The presynaptic axon terminal, if you look closely at it, they've drawn it here showing some swelling in that area where the vesicles dock. These vesicles are the things that actually contain the neurotransmitters. They're called the synaptic vesicles. And there's also a swelling drawn on here on the postsynaptic neuron, and those swellings are made up of various types of proteins, many of which are actually receptors for the neurotransmitters that will be released into the synaptic cleft and bind to the postsynaptic receptor. So those receptors present on the postsynaptic neuron will be bound by neurotransmitter release from the presynaptic neuron, cross that synaptic cleft, and then produce some response in the postsynaptic neuron, usually activate it or quiet it down so that it doesn't fire. Sometimes a presynaptic neuron can release more than one neurotransmitter, and if it does do that, the additional neurotransmitter is typically called a co-transmitter, and it usually does something to facilitate the binding of the primary neurotransmitter. It usually either helps it bind its receptor or it actually slows it down in terms of its binding to its receptor on the postsynaptic neuron. So presynaptic axon terminal, you see the little synaptic vesicles that store neurotransmitter. There's a synaptic cleft, and when these vesicles open up and release neurotransmitter, as you'll see in a moment, they travel across the synaptic cleft and bind to various sites on the postsynaptic receptor and induce some type of change in that postsynaptic neuron. Typically, as I said, make it fire or not fire. Let's go to the next slide now and see how these chemical synapses actually are very different in their morphology. This picture you see here is typically drawn in the textbook, but in reality they look very different, as you'll see on the next slide. Okay, take a look at figure 626 here, and it just shows the diversity that you can see in synapses. You saw figure 625, which shows the so-called typical synapse, but in reality these synapses aren't typical. They're very unusual looking things. And if you look back at figure 6.1 in this chapter and you look at the so-called typical neuron shape, and you see the cell body, the dendrites, the axon of the neuron, and the axon terminals, typically neurons, presynaptic neurons, synapse with a postsynaptic neuron on their cell body, and more specifically the dendrites of the postsynaptic neuron. So if you look back at figure 6.1, you would see presynaptic neurons synapsing with it, if it was drawn on there, with its cell body and its dendrites. In fact, you can actually see something that looks like that if you look at figure 6.5 in this chapter, that shows a presynaptic neuron, its axon terminals synapsing with the cell body and dendrites of the postsynaptic neuron. But it doesn't have to be exactly that way. A presynaptic neuron can actually synapse with the axon of the postsynaptic neuron. It can basically synapse anywhere with the postsynaptic neuron, but typically it's the presynaptic neuron synapses with the cell body and the dendrites of the postsynaptic neuron. If you look at figures B, C, and D in this figure 6.26, you can also see that these presynaptic terminals can be very diverse in their shape, as well as the postsynaptic cell body or dendrite. And the major reason for this is just to increase the amount of surface area or contact area that's made when these neurons synapse with each other. That allows them to more efficiently talk with each other, so to speak, by releasing their neurotransmitters and making lots of contacts with the postsynaptic neuron to tell it to do something or to not do something. These neurons, these synapses, by the way, are not static entities either. One of the beauties of the mammalian nervous system is that synapse is constantly changing their form and shape to more effectively communicate, to allow neurons to more effectively communicate with each other. So we refer to that as synaptic plasticity, which simply means that even though we essentially, after we're born, we essentially have the same neurons we're going to have for the rest of our life, there's very little evidence we make more neurons after we're adults. There's some evidence for that, but very little. But the reality is that the synapses that we have in our central nervous system can change, so that when one part of the brain needs to more effectively talk with another part of the brain, synapses can change. As I said, that's referred to as plasticity, and that's one of the reasons that our nervous system is so remarkable. This changing in the form of synapses that allows us to actually have a functional nervous system throughout our entire life. Now let's go to the next slide, which is figure 627, and look a little more closely at exactly what happens when neurotransmitters are actually released from the presynaptic neuron into the synaptic cleft and bind to the postsynaptic neuron. Okay, figure 627 shows a presynaptic neuron axon terminal synapsing with a postsynaptic neuron. And you've got a lot more detail in this figure than you did in figure 6.25. If you just follow the numbers here, if you look at number 1 that says action potential reaches terminal, this presynaptic neuron has fired, so it's generated an action potential. This action potential, as you know, typically starts up at the cell body around the dendrites and proceeds down the axon to the axon terminal. So now it's the axon terminal now is receiving the action potential. It's coming into the axon terminal. And this in turn, if you go to number 2 here, you see will actually open voltage gated calcium channels. So neurons are very specialized and they have predominantly voltage gated sodium channels in their cell body and along their dendrites and along the length of the axon. But in the axon terminals, they have these very specialized voltage gated calcium channels. So as the action potential reaches this site, it opens these voltage gated calcium channels because it's changing the membrane voltage down here. So calcium, if you go over to number 3 now, it shows calcium entering into the axon terminal through these voltage gated calcium channels. Now you go over to number 4, you see that there's a lot of these vesicles that contain neurotransmitters that are sitting in the so-called active zone of this axon terminal. These are the vesicles that are basically available to release neurotransmitter in response to an action potential. So when calcium enters into the axon terminal here, what happens is if you go down to the bottom of this figure to the left where you see the little vesicle and it's being bound by proteins called snares. So look at that figure down at the bottom left. These snares are proteins that are basically anchoring this vesicle with its neurotransmitter. Those little red dots are supposed to represent neurotransmitter to the plasma membrane of the neuron. You can see also that these vesicles have a protein associated with them called synaptotagmin. Synaptotagmin is not bound to the snare when you look at that figure on the left. But as calcium enters into the cell, into this axon terminal, that stimulates the process whereby synaptotagmin and the snares bind to each other. And that in turn initiates this complex process that causes the vesicle to fuse with the membrane of the neuron here in the axon terminal and forms an opening where the neurotransmitter can now leave the cell and enter into the synaptic cleft. So once that happens, that's actually drawn up above now as number 4, neurotransmitter release and diffusion, where the synaptotagmin and the snare have fused with each other due to the influx of calcium. The vesicle has fused with the plasma membrane of the neuron, and now it's dumping its neurotransmitter into the synaptic cleft. So in a moment, we'll look at what actually happens to the postsynaptic cell. You've got this neurotransmitter in the cleft here now, and that's going to bind to various types of receptors on the postsynaptic neuron, and it's going to cause something to happen to that neuron. As we'll see, it's either going to excite it or it's going to quiet it. Look at number 6 here, however. One other interesting aspect of neurons is that when they release neurotransmitter into a synapse, the neurotransmitter is in that synaptic cleft for a very, very brief period of time, less than a second, and then it's removed through various mechanisms. And one of the ways it's removed is just to have it diffuse away. Other ways that it's removed is there's often an enzyme in the cleft that will actually break it down very quickly, again less than a second. But another common way that the neurotransmitter is removed from the synaptic cleft is that it's reuptaken back into the neuron that released it. It's not drawn on here, but there's specialized proteins in the synaptic terminal here, the axon terminal. There's specialized proteins that are there that are not drawn in this figure that literally suck the neurotransmitter back into the axon terminal that released it, repackage them, and use them over again. So let's go to the next slide now. I want to show you a clinical example of a drug that takes advantage of this ability of axon terminals to literally suck back up the neurotransmitter they released through specialized reuptake transporters. This is a slide I wanted to throw in here called the clinical correlate of the CNS effects of cocaine. This figure in this particular PowerPoint is actually figure 6.34 from your book. We'll be looking at this again little bit later, but I threw it in here because it shows this process of neurotransmitter reuptake. If you look at this figure and you look at E and you see the arrows going out and back in, what that's supposed to represent is the fact that presynaptic neurons in their axon terminals, when they release neurotransmitter, as I said in the previous slide, one of the ways that they can remove neurotransmitter from the synaptic cleft is to basically reuptake it. And to do that, they use specialized proteins that are on the neuronal cell membrane here to pull the neurotransmitter back in, and typically they repackage it into a vesicle and use it again. Cocaine, which everyone is familiar with as a drug of abuse, it's a stimulant, and the way cocaine produces its effect is by taking advantage of these so-called reuptake proteins on presynaptic neurons. Cocaine's a plant alkaloid. It's made by the cocoa plant, predominantly in South America, and it works specifically on the neurotransmitter, predominantly, I should say, not specifically, but predominantly on the neurotransmitter dopamine. Now, there's certain parts of the brain that use dopamine a lot as a neurotransmitter, and one of these areas is a part of the brain that's involved in feelings of pleasure, called the nucleus accumbens, which is not relevant to this course, but that just happens to be an area of the brain in the limbic system that's involved in pleasure. So when a person uses cocaine and gets it into their bloodstream, it rapidly enters into the brain because it's very lipid soluble. So it's taken up by the brain, and it finds itself in these interstitial spaces here around neurons, including the synaptic cleft. Cocaine molecules would be here in the synaptic cleft if you were using cocaine. Well, when these particular neurons in the nucleus accumbens release dopamine, and that dopamine binds the postsynaptic dopamine receptors on the postsynaptic neuron, that process, because of the part of the brain that it's in, gives people the feeling of pleasure. So if cocaine is on board, it blocks the reuptake of this released dopamine so that it can't get back into the cell that released it. It's sitting on the dopamine reuptake transporter. It sits on there, and so dopamine remains in the synapse. It can't be vacuumed back up, and so it continues to stimulate postsynaptic receptors, and that produces excessive feelings of pleasure. So cocaine, an interesting compound that is made by a plant, has profound effects in the mammalian nervous system by virtue of its ability to bind to dopamine reuptake transporters in certain parts of the brain, and therefore cause dopamine to accumulate in the synaptic cleft in these regions and overly stimulate postsynaptic receptors and produce these tremendous feelings of pleasure. Let's go to the next slide now. Okay, this is figure 628 from Vander's 11th edition, and now we're going to start looking a little more closely at what happens at the postsynaptic cell. You've learned quite a lot at this point about presynaptic and postsynaptic neurons in terms of a synapse, and you've also learned just a few slides ago about this issue of convergence and divergence. And as you saw, if you went back and looked at that slide, that would be figure 6.24, you would see that in terms of convergence, you now know that neurons, basically a single neuron, can be receiving information from literally hundreds of different presynaptic neurons at the same time. And the information coming from those presynaptic neurons can be different. Some of them may be telling that postsynaptic neuron to fire, and some of them may be telling the postsynaptic neuron not to fire. And that neuron has to kind of sum up that information and decide whether or not it's going to generate an action potential. So when we're talking about the postsynaptic cell now, we're looking at a different type of receptor for the neurotransmitter. Now remember, up to this point we've been talking a lot about voltage-gated sodium channels opening up in terms of propagating an action potential. And action potentials propagating along an axon, getting to the terminal end of the axon, and then stimulating the release of neurotransmitter. So now we're looking at what that neurotransmitter that's released is doing at its receptor on the postsynaptic neuron. These receptors on the postsynaptic neuron are different from the voltage-gated channels we've been talking about, and pretty much exclusively sodium, voltage-gated sodium channels. These receptors on the postsynaptic neuron are usually in two different categories. One is called an ionotropic receptor, and the other is a metabotropic receptor. Now, the ionotropic receptors are basically receptors that are directly linked to an ion channel. And when the neurotransmitter is released and it binds to these so-called ionotropic receptors, it either closes the ion channel or it opens the ion channel, depending on the type of neurotransmitter. A metabotropic receptor, on the other hand, when a neurotransmitter is released into the synaptic cleft and it binds to these metabotropic receptors on the postsynaptic neuron, it doesn't directly open or close an ion channel. It produces some cascade of events that's usually involving G proteins that then, after a series of downstream events, can lead to the opening or closing of an ion channel. And remember, this issue of opening or closing ion channels is important, because in terms of whether or not a neuron is actually going to depolarize and generate an action potential has to do with making the inside of that neuron less negative. So therefore, we've learned a lot of ways that you can do that. How can you make the inside, how can you depolarize a neuron, i.e. make it less negative? One way is to send in positively charged cations via channels, and that would make the inside less negative. Another way would be to leak out negatively charged ions like chloride. That would make the inside of the neuron less negative and thus depolarize it towards the threshold of depolarization that it needs to reach before it actually fires and generates an action potential, which we've been talking a lot about. So what we're looking at here in this particular figure is what's called a so-called excitatory postsynaptic potential, or an EPSP. And all that means is that describes information that a postsynaptic neuron is receiving from presynaptic neurons that's telling it to fire, i.e. certain presynaptic neurons are releasing neurotransmitters that, through one of the mechanisms we've described, is somehow depolarizing this postsynaptic neuron and telling it to fire. Now, as I said earlier, the neuron has to weigh that information against information that it's receiving from neurons that are inhibitory and telling it not to fire, and sum these up and decide what to do. So at this point, you're looking at an EPSP here in this figure that's showing depolarization, but not to the point that it reaches threshold. And now let's look at the next slide that shows an IPSP, or information's being received by the neuron that's telling it not to fire. Okay, figure 629 here shows an IPSP, or an inhibitory postsynaptic potential, meaning that at the postsynaptic neuron, neurotransmitter is binding receptors that somehow leads to the interior of that postsynaptic neuron becoming more negative or hyperpolarized, as you can see here, which means it's going to be less moving away from threshold, meaning that it's going to be even less likely to fire or generate an action potential than it was when it was at its resting membrane potential of around negative 70 millivolts. Now at this point you ought to realize that the way you could make the interior of a postsynaptic neuron more negative would be if the neurotransmitter that was released by the presynaptic neurons bound potassium channels on the postsynaptic neuron. Potassium being a cation, positively charged ion, and being present in much higher concentrations inside of cells, neurons, and other cells for that matter, than it is on the outside. If you have a neurotransmitter binding potassium channels on the postsynaptic neuron and opening up those channels, then potassium would be flowing out of the neuron down its concentration gradient. So you would, in essence, have positively charged ions, potassium, leaving the interior of the neuron. And of course, that would make it more negative in the interior. That's one way to do it, to make a postsynaptic neuron more negative and generate an inhibitory postsynaptic potential. Another way, of course, common sense dictates that if your neurotransmitter from your presynaptic neuron bound receptors on the postsynaptic neuron that opened chloride channels, then if chloride flowed into the cell, chloride having a negative charge, chloride ions are anions, of course, then that would make the interior of the neuron more negative as well and hyperpolarize it like you see here and produce an inhibitory postsynaptic potential. And in fact, in those cells that actively regulate intracellular chloride concentrations via active transport out of the neurons, the chloride equilibrium potential is more negative than the resting potential. And basically what that means is if a neurotransmitter binds to the postsynaptic receptor and does in fact open chloride channels, then chloride will in fact flow into the cell and produce a hyperpolarization like you see here. Either way, what you need to understand from this particular PowerPoint and the previous one is that, again, postsynaptic neurons are constantly receiving information of varying types from the presynaptic neurons that synapse with them, and some of this information via neurotransmitter release is hyperpolarizing the postsynaptic neuron and telling it not to fire, and some of this information in the form of neurotransmitters from the presynaptic neuron is telling this postsynaptic neuron to fire by hypopolarizing it or depolarizing it. And in the end, this neuron has to sum up that information and decide whether to fire or not fire. So we'll look at synaptic integration next and see exactly how that happens. All right, in the hundreds or even thousands of presynaptic neurons that are releasing neurotransmitter at any given time and influencing a single postsynaptic neuron, the membrane potential of that postsynaptic neuron at any particular moment is the result of the influence of all these inputs from the presynaptic neurons, and which you've seen in the last few slides, some of which are inhibitory and some of which are excitatory. So to depolarize the membrane and actually get an action potential, there has to be a time when the excitatory input predominates, and to keep it from firing or to make sure an action potential doesn't occur, there has to be a time when the inhibitory postsynaptic inputs predominate and keep the neuron quiet. And this has to do with what I've been alluding to earlier in terms of summation, when a neuron can actually sum up the information it's receiving and make a determination as to whether or not it's going to fire. And again, that's going to have to do with whether or not it's able to depolarize itself to the threshold level and actually start the opening of voltage-gated sodium channels like we discussed previously when we talked about how we generate action potentials. So if you look at this figure 631, this is supposed to represent a neuroscience experiment where basically on your left you see this single postsynaptic neuron, and it's got two excitatory inputs coming to it, labeled here as A and B, the green excitatory inputs, and it's got one inhibitory input coming to it, the red neuron, presynaptic neuron. It's labeled as C, and there's a microelectrode stuck in this thing to record the membrane potential, and that's what's drawn in the gray box on the right. And again, theoretically, the scientist has each of these two excitatory and one inhibitory presynaptic inputs are wired so that they can push a button and get them to fire when they want them to. So in this unrealistic experiment, you couldn't really do this experiment that I'm aware of anyway. On the first panel, labeled panel 1 there, what they've done is just shown two distinct non-overlapping graded depolarizations from input A. Again, that's an excitatory input. So what they've done there is they stimulated input A. It released some type of excitatory neurotransmitter, which typically means it opened ligand-gated sodium channels on the postsynaptic neuron. Sodium flowed into the cell, and it depolarized, but it wasn't enough of a depolarization to bring it up to threshold and make it fire. The membrane potential was allowed to return to the resting potential, and then it was stimulated again. So you just have two separate stimulations from input A here, an excitatory presynaptic neuron that depolarized this postsynaptic neuron here, but not enough to actually make it reach threshold and fire. So in panel 2 now, if you move to the right, you see two overlapping graded depolarizations, which demonstrates what's called temporal summation. In this case, they stimulated the excitatory input A, but before, and it depolarized the cell, but before it returned to its resting membrane potential, they stimulated it again. And so now, at that particular moment, when you have the second stimulation, you have more open ion channels in that postsynaptic membrane than you did in panel A. And again, let's just, for simplicity's sake, assume these ion channels are sodium channels, because that very often it is when it's excitatory. It's opening sodium channels, and sodium's flowing into the cell, making it more positive and depolarizing it. So in this case, temporal summation. They call it temporal summation because the input signals arrive from the same presynaptic cell but at different times. So this is just presynaptic neuron A stimulating this cell at different times, and in this case the second stimulation occurred before it returned to resting membrane potential from the first stimulation. So now move to the right and look at panel 3, and this is an example of spatial summation, where you have distinct actions of stimulating neurons A and B, and in this case they first stimulated with B. Okay, remember, if you look to the left there, B is also an excitatory presynaptic neuron, so it also released an excitatory neurotransmitter. They depolarized the neuron, it returned to the resting membrane potential, and then they stimulated A and B at the same time. And so your depolarization that you got at that point was slightly greater than it was when you just used presynaptic neuron B. And again, that's because you've got two presynaptic neurons now firing at the same time and both releasing excitatory neurotransmitter, which depolarizes this postsynaptic neuron even more than either one alone can. This is called spatial summation because the two inputs occurred at different locations on the same cell. So you have A and B stimulating at different spots on the postsynaptic neuron here. So that's called spatial summation as opposed to temporal summation, where you have overlapping summations, but it's coming from the same presynaptic neuron. So finally you go to panel 4 here, and you see that A and B are stimulated enough now to cause a superthreshold graded depolarization, which is just a fancy term meaning that you've got input enough from all of these excitatory presynaptic neurons. Remember, both A and B are excitatory, and you're firing them both in both temporally and spatially, and that results in enough opened ion channels. Again, let's assume they're sodium channels, to now let enough sodium flow in to depolarize the inside of that cell to whatever the threshold is. Usually, typically it's around -15 to 0 millivolts, and that generated an action potential. So that it basically summed up, using spatial and temporal summation, excitatory inputs from A and B, and was able to generate an action potential. And this is actually what's occurring all the time in neurons, where spatial summation and temporal summation are occurring, and at any given time you might have a situation where excitatory presynaptic neurons are summed up at just the right time and just the right number to open up enough ion channels that allows positively charged ions, typically sodium, to enter the cell to cause it to reach threshold level of depolarization and fire. And finally, the last panel just shows that basically, if you read your text, you'll read that negative presynaptic inputs can basically do the same thing. In other words, you can have temporal summation or spatial summation of negative presynaptic inputs as well, just like you can with excitatory presynaptic inputs. But they just show a simple example here of if you had at any given moment a single excitatory presynaptic input, that would be A here in this last panel, number 5. And an inhibitory presynaptic input, C, firing at the same time as you do on the right-hand side of panel 5. You see that there's basically no change in the membrane potential because they cancel each other out. So this is just a fairly simple figure to show you this concept of summing up information that a postsynaptic neuron does, both temporal summation and spatial summation, and information coming from both inhibitory and excitatory presynaptic neurons, and how it sums this information up, and whether or not the postsynaptic neuron is able to actually depolarize to the threshold level and generate an action potential. All right, let's go on to the next slide now. Okay, figure 632. Figure 632 is simply showing the direction of current after an excitatory synapse, which is the example on the top of this figure, an inhibitory synapse, which is the one on the bottom in this figure. If you look at the top one, you see that the direction of current is away from the synapse, and this should make sense because remember the top one is the excitatory synapse, meaning that particular neuron where the synapse is occurring, that postsynaptic neuron is being depolarized. In other words, the inside of it is being made more positive, and we've talked about this before, this process of depolarizing and generation of an action potential. And in that figure you can see the positive charges accumulating there and moving towards the initial segment on the right there. And the initial segment is a part of the neuron that has lots of voltage-gated sodium channels. So if the excitatory input here at the excitatory synapse is great enough, and there's enough movement of these positive charges towards the initial segment, enough movement of current towards this initial segment, it could actually end up in this neuron generating an action potential. On the other hand, the inhibitory synapse on the bottom here, in this case, you can see positive charges moving towards the actual synapse as opposed to away from it, because remember, this is an inhibitory synapse, meaning that this neuron, or these neurons that would be here, are releasing neurotransmitters that are binding to this postsynaptic neuron that you're looking at, and causing it to become hyperpolarized or more negative interiorly, and that's dragging positive charges towards this site and hyperpolarizing the neuron and making it much less likely to be able to generate an action potential. Okay, let's go to the next slide now. Okay, figure 6.33 shows an example of axo-axonal communication. In this particular example, it shows B as a presynaptic neuron that's releasing neurotransmitter into a synaptic cleft that's affecting its postsynaptic neuron, which in this case is labeled as C. And that's kind of the classic mechanism we've been looking at up to this point, where you have a single presynaptic neuron releasing neurotransmitter and affecting postsynaptic receptors on a postsynaptic neuron. Well, in this situation, we've got a little bit of a complicating factor. In this situation, in axo-axonal communication, you have another neuron, in this case labeled as A, that's actually synapsing with the axon terminal of neuron B. So A in this particular figure is not directly affecting the postsynaptic neuron here, C. It instead is affecting the presynaptic neuron B and its ability to communicate with the postsynaptic neuron C. In other words, the neuron labeled A here is releasing some type of neurotransmitter that's binding to receptors that are present on the axon terminal of B, and depending on what that neurotransmitter is, it could be influencing neuron B to release less neurotransmitter, which in turn, of course, would affect its ability to communicate with the postsynaptic neuron C, or it may be releasing a neurotransmitter that's affecting the neuron B, that's causing it to release more of its neurotransmitter, which of course in turn would also affect the way that it communicates with neuron C. So this axo-axonal communication here just shows you how complicated synapses can get, where they're never simplistic, and in this case it's showing that a presynaptic neuron B can have multiple influences on it, as you're seeing here. And depending on the type of presynaptic receptors that are activated by the neurotransmitter that's released from neuron A in this particular case, the presynaptic effect may decrease the amount of neurotransmitter released from B, as I alluded to before. And if it does, it's called presynaptic inhibition. Or, as I also said, it may increase the amount of neurotransmitter released by B, and if it does that, it's called presynaptic facilitation. So once again, you see that these synapses are very complicated things, and there's multiple inputs. In this case, it's showing other neurons affecting presynaptic neuron B and its postsynaptic receptors on postsynaptic neuron C. Okay, let's take a look at figure 634 here. This figure has a lot of clinical relevance, as you can imagine, because up to this point you've learned a lot about neurons, synapses, postsynaptic receptors, presynaptic autoreceptors, etc., etc. And as you can imagine, over the years, drugs have been developed to affect many of these things that we've been talking about up to this point, central nervous system and peripheral nervous system drugs. So as you look at this figure, for example, look at what it's telling you here. It says possible drug effects on synaptic effectiveness. So many aspects of synaptic functioning can now be modified by drugs. For example, release and degradation of the neurotransmitter inside the terminal. For example, a drug like reserpine blocks the uptake of norepinephrine, serotonin, and dopamine into vesicles inside the presynaptic neurons where it's normally stored. And when it's stored in those vesicles, it's protected from enzymes that degrade them. So if they're unable to be stored in the vesicles because a drug like reserpine is around, it makes those neurotransmitters susceptible to degrading enzymes, and subsequently the nerve terminal will release less neurotransmitter because it can't store it into vesicles. Another example, increased neurotransmitter release into the synapse. Certain types of amphetamine, especially synthetic amphetamine, through various mechanisms can increase neurotransmitter release into the synapse, particularly dopamine. Prevention of neurotransmitter release into the synapse. Your book uses an example in terms of disease, tetanus toxin. Tetanus toxin that you get from the bacterium Clostridium tetani is a protease that destroys those snare proteins we talked about previously in the presynaptic terminal, so that you can't get that fusion of vesicles with the membrane of the presynaptic terminal, and therefore you don't get neurotransmitter release. So tetanus toxin is an example of a substance that can prevent neurotransmitter release into the synapse. Inhibition of synthesis of the neurotransmitter. Well, drugs do that as well. There's a drug called mupirocin that actually inhibits the hydroxylation of the amino acid tyrosine to dopa, which is required in the pathway to make dopamine. So by giving an individual mupirocin, you inhibit their ability to manufacture the neurotransmitter dopamine. You also have drugs that reduce the uptake of neurotransmitter from the synapse. Earlier in this chapter, I gave you an example of an illegal drug that does that, cocaine, which blocks the reuptake of dopamine in synapses in the limbic system in the brain and produces the effects of euphoria that are seen with that drug. more pharmaceutical examples are the specific serotonin reuptake inhibitors that we use to treat depression, such as fluoxetine and paroxetine and citalopram. Those all block specialized proteins that are on the presynaptic membrane that are involved in reuptaking the released neurotransmitter and bringing it back into the neuron that released it, repackaging it, and using it over again. We've taken advantage of the knowledge of that by manufacturing drugs like the SSRIs. There's drugs that reduce the degradation of the neurotransmitter in the synapse. We have lots of central nervous system and peripheral nervous system agonists and antagonists. For example, L-DOPA in the treatment of Parkinson's disease is a dopamine agonist, so it works at postsynaptic neurons on dopamine receptors to mimic dopamine. Naloxone is an example of a drug that's an antagonist that blocks opiate receptors so that opiates cannot bind their receptor. It sits on the opiate receptor and doesn't allow the endogenous opiates like the endorphins to sit on the opiate receptor. And of course it blocks the ability of exogenous opiates like heroin that somebody might take to sit on the opiate receptor. Naloxone itself is not an agonist. It doesn't activate the receptor. It just sits on it so the agonist cannot get to it. So when you look at this picture, 6.34, just keep in mind that it's this kind of knowledge, all these things that you're seeing here in terms of these issues of manufacturing neurotransmitter, storing neurotransmitter, releasing neurotransmitter, degrading neurotransmitter. We have all kinds of pharmaceutical drugs that mimic these processes for several different neurotransmitters. And again, the issue of agonists. So you have acetylcholine agonists, we have dopamine agonists, we have serotonin agonists, etc. These are all things that you'll learn about in a pharmacology course, but we kind of present them here. I'm talking about them here simply because I want you to see them in the context of a presynaptic and postsynaptic neuron and all the issues that are associated with normal neurotransmitter release. All of these things can be affected by several different types of drugs, and it's information that'll be very useful to you later when you do have a pharmacology course, if you haven't already had one, in terms of understanding how central and peripheral nervous system drugs produce their effect. All right, let's go to the next slide.